Assalamualaikum and hi. We are from Group 3. We'll be presenting about the detections of SARS-CoV-2 protein by Western blotting technique. Principles of Western blotting, which is Western blotting is a method to separate protein according to their size, and then the protein of interest were detected with the specific antibodies. Firstly, the protein sample are separated by SDS polyacrylamide gel electrophoresis or SDS page to separate various protein contained in the sample according to the size, charge, or other differences in individual protein bands. Next, the separated protein then transferred to the membrane, commonly used nitrocellulose or PVDF membrane, through a process known as a blotting, using a method either semi-dry transfer or wet transfer. Third is the blocking solutions, solutions used to prevent non-specific binding antibodies to the membrane, like a bovine serum albumin and the skim milk. The membrane will transfer protein, then probe with the primary antibody, proceed with the secondary antibody labeled with an enzyme such as horseradish peroxidase. Then the bound enzyme activity then used to detect the target protein, visualized by the chemiluminescence or chromogenic method. Then after the color development happen, the patterns of the separated protein is captured by Western blot gel imager, and compare the band positions to the protein ladder, we can estimate the size of the protein. Next, we move to the method of the Western blotting, particularly in detecting SARS-CoV-2 protein. Firstly, the cells that are transfected with the plasmid encoding spike glycoproteins of SARS-CoV-2, SARS-CoV, MERS-CoV, and MHV were lysed at 40 hours post-transfections by Ripa buffer. Then the virus supernatant were centrifuged at 25,000 rpm for 2 hours in a Beckman SW41 rotor at 4 degrees Celsius through a 20% sucrose cushions in order to pellet down the pseudovariants. Then the virus supernatant again centrifuged at 25,000 rpm for 2 hours at 4 degrees Celsius through 20% of sucrose cushions, and the virus pellets were resuspended into 30 microliter Ripa buffer. Then the sample boiled for 10 minutes and then separated in 10% SDS page gel and then transferred to the nitrocellulose filter membrane through a process known as blotting. After it was blocked by 5% milk, the membrane were bloated with primary antibodies, followed by incubated with horseradish peroxidase, which is a conjugated secondary antibodies. Then it was visualized with the chemiluminescence reagent. Then, as a result, MHVS protein were detected using polyclonal good anti-MHVS antibody, and then the SARS-CoV-S protein bloated with either polyclonal anti-SARS-S1 antibody T62 or mouse monoclonal against SARS-S1 antibody MMO2, and then the MERS-CoV and SARS-CoV-2 S protein were detected using the mouse monoclonal anti-MERS as and anti-flag M2 antibody, respectively.
This slide shows the advantages of the Western blotting. First, Western blotting is an excellent method with high sensitivity and high specificity. Why Western blotting is high sensitivity? Because it can detect as little as 0.1 nanogram of protein in the sample. And why Western blotting is high specificity? Because in this technique, the specific antibody will show the affinity for the specific protein. Thus, this process also can selectively detect a target protein even in a mixture that contains the different type of the proteins. Next, Western blotting also can separate the protein according to their molecular weight. So basically, we will use the gel electrophoresis such as SDS page in order to sort the protein according to their molecular weight. Then, we will see the separation of the protein band on the SDS page, where this protein band then will be transferred to the membrane such as nitrocellulose, so that the detections of the specific protein can be done better. And since the Western blotting detects the protein by their size and ability, to bind to the antibody, it is appropriate for evaluating the protein expression in cells and further analysis of protein fractions during the protein purification. Next, limitation of Western blotting. First, the process is time-consuming and complex. Why it is complex? Because we need to separate the protein first, and then we need to transfer the protein to the membrane, and we also need to use the antibody in order to detect the specific protein. So, since this process is complex, then it must be performed by the well-trained personnel. Next, it is a very sensitive process, so any imbalance in the process such as the protein isolation, buffer, type of separation, and also gel concentration actually can affect the result of the entire process. The techniques requires first antibody, and it is a costly process with the cost of the antibodies and also expensive methods. Last one, some antibodies might exhibit off-target effects by interaction with more than one protein in the sample. I think that's all from my group. If you guys have any questions about this topic, then you may ask later, okay? Thank you and Assalamualaikum warahmatullahi wabarakatuh.